Hi there. We are completing the second half of Chapter 5's Antibody Structure and Function lecture. And we are picking up right where we left off on the three-dimensional structure of antibodies. We've been looking at antibodies throughout the lecture and throughout the semester so far as a 2D structure shaped like a Y, but it's not. Clearly, it's a 3D structure that's sort of semi-reminiscent of a Y. If you see, here's the two regions that would represent the top of the Y, and then it comes straight down and sort of represents the bottom of the Y. But in fact, these are very conformational shapes and not 2D at all. The structure is, of course, formed by those four basic chains that we had talked about. The two heavy chains that are the isotypes, the amino acids that make up each of the unique heavy chains, either the amino acids that make it an alpha chain, delta, gamma, epsilon, or mu. And then obviously we have the two light chains as well. The light chains are either kappa or lambda, as you recall. And so these chains are, they don't exist in kind of a linear fashion. Instead, what happens to the chains is they fold on each other in what we call a beta pleated sheet. And what beta pleating is I don't know if you've ever seen or made an accordion pleat, where you fold you fold a piece of paper one direction, then the other, then the other, then the other. People use it to make like tissue flowers, tissue flowers, or paper fans. Those are accordion sheets, and that's what we're seeing here when we're making those peptide chains of the heavy and light chains into this beta pleated sheet. So we're calling that pleating a beta pleated sheet. And when we make that pleating, we're making one domain here, or one region here. We're making one region here. Combined, they make the variable region. And then we have similar pleating down the bottom in the constant region. I think it's a little easier to illustrate what's happening to the chains when we look at it this way. You see it goes down, then up, and then down, and up, and down again. I think I missed someplace, up and around and down. So it's making a pleating. I'm going to take these off now so that we can see the rest of the structure as we go along. Those are the beta pleated sheets. And those beta pleated sheets are held in place by those disulfide bonds that we talked about before. The disulfide bonds are going to hold that pleating in place and make it a stabilized structure for both the variable and the constant domains. These globular regions are almost, if you think of them almost like a balloon shape at the end. Within the variable region, we talked about that being the site of antigen-antibody binding. And in fact, we talked about it like it being right on the tip of the variable region in our little Y-shaped 2D picture. But what in reality happens is that within these two kind of globular regions, there's something we call the immunoglobulin fold or the immunoglobulin barrel. And what happens is that antigen gets captured within the barrel, binding to a really small sequence of amino acids that are caused by that memory, that introduction to the antigen, whether it's processed through the MHC or it's directly found and processed through B cells. But in any event, that's that really variable region that's made just for that specific antigen. And there's a small group of peptides within the middle of these two balloon structures. And as I said, we call that the immunoglobulin fold or the immunoglobulin barrel. Those very small regions of amino acids that are going to actually bind the antigen in are called the hypervariable region. We call them that because that's the area of the exact match of what we're creating to bind to the antigen and of the antibody. So hypervariable region is the site of actual immunoglobulin antigen binding within this immunoglobulin folder barrel. Imagine there being a little place in here that you can bind in an antigen. I'm trying to remember if I have a picture. I don't have another picture. When we look at the rest of this structure, we're looking at where do we see that genetic diversity that we've been talking so much about in the creation of antibodies primarily through MHC processing, but also again through that during B cell development, the production of genetic variations in B cells in order to be able to identify and bind these myriad of antigens that come along. And in fact, antigenic diversity is found in the variable region that we had talked about. It's found in what are these CDR regions, CDR1, CDR3, and CDR2. There's a total of 6 CDR regions per antigen. Because there are 3 regions, 1, 2, and 3, and there's a heavy and a light chain for each of those regions. It's not illustrated great here, but obviously each one of these represents a heavy and a light chain because we know the variable region has heavy and light chains. So there's an opportunity, there's 6 opportunities to create these really antigenically diverse regions for the antibody. It takes just that teeniest little change in amino acid structures within those regions to create conformational changes for different kinds of antigens. So that's really how, when we're talking about antigenic diversity, we're talking about making small amino acid changes within both heavy and light chains of each of these 3 different regions. And by mixing it up, we get great antigenic diversity. Please let me know if this doesn't make sense to you. I know it's a tough concept, especially when you don't have the opportunity to ask a question. So feel free, please ask me a question if you have any questions. I'm going to move into the slideshow now that I don't have to take any additional working parts off of the slide deck. Before we move out of the structure of antibodies and into each of the individual antibody classes, let's take a look just a little bit further to look at the antibody structure, what the activity in each one of the regions means in terms of functionality of the antibody, the immunoglobulin. Antibodies were discovered to be able to be treated with enzymes, and we used a couple of them to really have scientists and researchers reveal the components of that Y-shaped structure that we've seen. 2 enzymes get used, two types of proteolytic enzymes get used, and what happens is they cleave or break the immunoglobulin into different fragments. The enzymes that we used and we're going to look at, one is called papain and one is called pepsin. And the fragments that we see resulting from the cleavage, the first one is called the FC fragment, or the fragment crystalline. The second is the FAB, the fragment antigen binding. I think we can imagine where that's going to show up. And the third is the FAB2 fragment, or the fragment antigen binding 2. You're going to see in a minute that it's actually breaking down this FAB fragment in a different way. So let's look at the actions of the 2 enzymes that were used, papain and pepsin, and then look at how that relates to the immunoglobulin structure that we recognize today. So we have our Y shape, again, just for the sake of being able to use the structure, the two-dimensional structure for our discussion. They discovered that the enzyme papain could break down the disulfide bonds, specifically the disulfide bonds at the hinge region only. So this enzyme cleaved or broke, so cleave comes from the word cleavage, which is to break. So it breaks the disulfide bonds in the hinge region. And it resulted in 3 fragments. 2 are in the FAB region, so we have 1 and 2. And you see these, the heavy chain and the light chain are still bound by disulfide bonds, right? They're still bound together. So we have one FAB fragment, 2 FAB fragments, and then that fragment crystalline or crystallizable, depends on where you read it, the 3rd region. So that's the FC region. And that represents really those heavy chains that are in the constant region. What do each of these fragments do? The FAB fragment, obviously, and it makes sense, is named because that's the place where it has the antibody that which we abbreviate as AB, right? So antibody we abbreviate as AB. I'm looking at the F and it keeps saying AB. Antibody is abbreviated as AB. So the fragment of the antibody region is the FAB fragment. So FAB fragments, there are 2 of them, consists of half of the heavy chain. So here's the heavy chain, and here's the other half, right? So we get half of the heavy chain and 1 full light chain in each of these FAB fragments. The heavy and light chains are still held together by disulfide bonds, and this is going to contain that variable region. We recognize it here, but remember we talked about it, that in reality that's the site of the antigen-antibody binding site, the immunoglobulin fold, is held within here. So the FAB region holds the immunoglobulin fold, still held together by disulfide bonds, and it's the variable region. The FC fragment, on the other hand, is the fragment crystalline, and it reflects the major portion of the antibody's constant region. If you remember, there was a little teeny bit of the light chain that also had a constant region. It's not illustrated as well here as it was on some of our earlier slides. But the FC region really isn't the site of antigen-antibody binding. It represents half of the 2 H chains that are also still held together by disulfide bonds, 2 sulfide bonds, disulfide bonds. This is an important factor, and we talked about it last week, how this becomes important, we talked about it in the first half of the chapter, how this portion, even though it doesn't bind antigen to it, it's important because it will, it's going to be able to be recognized by other cells that have a receptor for this region of the constant region, and it's going to result in that opsonization, that coating. So if we imagined when we showed it, we had the pathogen binding here, right, and we bound the antibody in here, and this end was sticking out, and that we could come along and have another cell here bind to this region, this constant region, with receptors on another cell, and that is how we trigger additional phagocytosis, for example, is by recognizing that this is sticking out and because it's stuck to another cell, essentially. This we can also see that this is going to play an important role in a couple a couple of lectures from now when we talk about complement. So this FC region, though it's not the variable region, it's not the site of antigen-antibody binding, is important in its structure for the function of the immune system. So let's move on. So that was the papain cleavage model. And we ended up with 2 FAB and 1 FC. So now let's look and see what happens if the enzyme pepsin digests the antibody. We end up with different molecules. Unlike the FAB from papain digestion, when we use pepsin as the enzyme for cleavage, we end up with, instead of two individual FAB regions, we end up with the 2 FAB regions, but still bound together by disulfide bonds. So pepsin cleaves immunoglobulins at a different place than papain does. This is important and actually quite helpful to us in the laboratory when we're using different tests, because we've maintained a very large structure with a couple of areas for antigen binding. So we'll see that this model is able to be used in some of the lab testing that we discuss. But in any event, we end up with one FAB2 region. So we end up with one FAB2 molecule. And as I said, in the lab, it has the ability to bind to antigen, and some of the reactions that we use, and we're going to talk about in a couple of lectures from now, are agglutination precipitation reactions. So this is a good thing. This model of cleavage is very helpful to us. The FC region, you can see it has a little apostrophe after it because these are not exactly the same as our other FC region. In this case, pepsin cleaves off all of the bonding, and you end up with very small little particles of the FC region in pieces. Those are not quite so helpful to us in the laboratory, but that's the way the enzyme works. So you can see from those two illustrations that scientists and researchers were able to identify different parts of the molecule, and then were able to do testing to identify their functionality. So let's move on to, kind of the big prize of the lecture, antibody production and the 5 classes. Yes, indeed, there are still 5 classes of antibodies. Let's look at each one of them individually and structurally. So this again is an illustration of each of the 5 antibody classes. 3 of them take on sort of the traditional Y shape that we see. They all have the same basic structure. It's just how we use them and put them together to make an antibody. So IgG, IgE, and IgD are said to be in monomeric or monomer, M-O-N-O-M-E-R, monomer structures. That means one structural unit of the Y shape. So we have the Y shape and it's one structural unit. We call that a monomer. IgA is a dimer. Here's one structural Y and here's another structural Y. So there's 2 structural Ys, so we call it a dimer 2. IgM is what we call a pentamer. There's 5 monomeric units, and they are also held together. They're held together by a special chain. We see here on IgA, we see this special little joining chain that holds those 2 units together into a dimer form. In fact, and we're going to see in just a moment, IgA actually can be seen in a monomer form. It's just not as common. And IgM, we hold them together as well with another. You can see here's another joining chain. They hold them together as well. Joining chains hold them together. Those joining chains are actually quite important in the function of these 2 molecules. You'll see in a few minutes. So that's the 5 antibody classes and their basic appearance. So let's start with, the granddaddy of all of them, IgG. IgG it's probably a misnomer to say it's the most important, but it's the most abundant for sure. 75 to 80% of all the serum immunoglobulins are IgG for a number of reasons. Number one, IgG is what's produced when we have secondary immune responses. That's second exposure. IgG is not exclusively in the tissues or the vasculature. It's found intravascularly and extravascularly. It's that single Y-shaped unit, that monomer, and we know that it has gamma heavy chains. We also know that IgG has a very nice long half-life. And again, when we talked about second exposures and creating memory, that the immunoglobulins that get produced stick around for a long time, and the beta cells, I keep saying beta, the B cells that are producing them are sticking around for a longer period of time. They're watching out to make sure that we don't encounter that pathogen again. So the half-life is actually quite long, 23 days. So all of that together, the fact that it's the primary immunoglobulin of what we call the secondary immune response, or the second time we see an antigen, that memory response, combined with the fact that you can find it intra and extravascularly, and it's got a nice long half-life, all those three things combine up to make it a very abundant immunoglobulin of the immune system. What kind of functions do we have for this? Well, we've talked already about that it provides immunity, for passive immunity for the newborn, because IgG can cross the placenta. We talked before about how it can bind complement, which is a process where particles like, bacteria or actually any pathogen that IgG can bind to, that has an antigen IgG can bind to, will trigger an immune response called complement. We're going to spend an entire chapter on complement, so I'm not spending a lot of time on it, but the goal of complement is cell lysis. We're looking to kill whatever that pathogen is. What else can IgG do? It can, as I said, it can coat the pathogen antigen to enhance phagocytosis, and we've talked about this word a couple of times, opsonization. It's an enhanced phagocytic process. And IgG on its own, when it binds to antigens of various pathogens, toxins, viruses, bacteria, it can neutralize it. It can actually cause the destruction of that pathogen. So lots of good things come from IgG. In the laboratory, we use IgG with a fairly high degree of regularity. In the laboratory, we can there are various kinds of testing. The first ones, the most basic ones, are called precipitation or agglutination reactions. Precipitation is one where particles fall out of solution, sort of if you mix sand and water and the sand falls to the bottom. That's a form of precipitation. In this case, it's not. It's really involving soluble particles, things that are dissolved, but the concept is the same. Agglutination, on the other hand, the second kind of laboratory testing that we use in this first step, is clumping. It's more like a clumping. I think you guys all looked at blood typing. Blood typing is an agglutination reaction where the red blood cells clump together when antibodies and antigens bind together. So in vitro, these are in vitro reactions. These don't happen in the host. These are in the laboratory. So in vitro means, in the lab. In vivo is in the body. So IgG is used for really a couple of different kinds of lab testing, agglutination and precipitation reactions. IgG, we're going to see why. IgM is better at agglutination and IgG is better at precipitation, but it can be used in both. And we alluded to just a second ago, and in our first lecture on this, that there are 4 subclasses of IgG molecules. 4 subclasses: IgG1, IgG2, IgG3, and IgG4. How they vary is really in the hinge region. If you look at the illustration, this is a great illustration of the hinge region of all of them and how they vary. They have different conformational structure in the disulfide bonds, particularly at the hinge. Variations to this end up resulting in the ability of IgG to be able to respond to different kinds of pathogens. So for example, IgG1 and IgG3 are really strong responders to antigens that are protein based, those complex protein antigens or epitopes, right? IgG2 and IgG4 respond really well to those simple carbohydrate, polysaccharide kind of antigens. All of these subclasses can respond in what we call the secondary immune response. We're going to review that in just a minute, but keep that in mind. All 4 of these are all able to respond equally in secondary immune responses. Let's move on to the alpha chain. IgA, or immunoglobulin with alpha chains, are really interesting. IgA is found primarily in secretions of the host. Things like, we talked about the very first week, MALT, that mucosal associated lymphoid tissue. They're found very heavily in those kinds of areas, respiratory, urinary bowel areas where there's mucosal areas. We know it has alpha chains that are the heavy chains. And it's important to note that we can have this in both a monomeric, monomer, or dimer forms. So it can be either in that 1Y shape or the 2 Ys kind of back to back. So here's 1 Y and here's another Y. The dimer form is held together by this chain. It's not really a J, it's sort of a, well, it's kind of like a little loop like that. But they call it a J chain. That's why it's illustrated exactly like a J. We're going to see in just a second, this is a really important immunoglobulin for any place where there's a mucosa, respiratory, urinary, bowel. And what makes this important, do you see this little chain right here? This we call the secretory component. Secretory. Because it's going to be involved in areas of secretions. So we'll take a look at that, how that process works in just a minute. So the structure of a dimeric IgA is 1 monomer, 2 monomers held together by a joining chain that has a secretory component that runs across it. That monomeric version does not have a J chain. It doesn't have a secretory component. So be careful. They definitely have different structures and different purposes. IgA represents about 10 to 15% of the total immunoglobulin pool. Important notes about IgA: it does not cross the placenta. It does not bind complement. But where did we see it? It was found in very large quantities in breast milk, which transfers immunity to the gut of an infant. So that's a good thing. That's going to transfer some of that passive immunity to an infant. As I said, there are 2 subclasses of IgA. The one we're mostly concerned about is IgA2, but let's look at IgA1 really quickly. The IgA1 and IgA2 differ, in content just by a few, 10, 15, maybe 20 amino acids, actually 22 amino acids difference, but only about 13 or so are in this, in the hinge region. So we have a slight variation in the hinge region of IgA1 and IgA2. What the IgA1 mainly does, it's mainly found in the, we say serum, or in the lab it would be serum. Obviously in the host it would be in the plasma, so it's in the plasma of the host. And what it does is it downregulates or minimizes phagocytosis that's been triggered by IgG. So it's one of suppression more than activation. IgA also can cause some of that chemical calling, that release of cytokines, that chemotaxis. It can have some evidence of, there's evidence of being able to kill bacteria. And of course, because we just talked about chemotaxis, we have some cytokines that get released in both killing bacteria, the bactericidal cytokines, as well as those chemotactic cytokines. So IgA1 is very useful as an anti-inflammatory reagent, but not so much what we think of as the real purpose of IgA. IgA2, the subclass IgA2, is much more prevalent, found mainly in secretions of the mucosa. As I said, it's a dimer, and what it does is it actually blocks the antigen from being able to penetrate further into the body. These initial defenses against pathogens are really important. And we're going to see in just a second how this happens. So it's a really important part of sort of the innate immune system, that initial immune system of trying to keep a pathogen from entering our body. As I said, it has this secretory component that helps it become more resistant to bacteria, but we're going to really see in a second how the secretory component aids IgA in the process of sort of patrolling the mucosa. This illustration is in your book, I believe. But you can see here, and I hope you can see here, IgA, just like every other immunoglobulin, is produced from plasma cells. So there's a plasma cell with its eccentric nucleus, and oh look, there's a couple of monomers of IgA. Oh, and what else are they producing here? They're producing J chains right there. That's that little J chain. These plasma cells are generally found in MALT, the mucosal-associated lymphoid tissue. They get produced in parts, and they end up being released from the plasma cell in its dimer form. So that's the first stage. The IgA dimer notice it doesn't have that secretory component yet. The secretory component is actually on the surface of epithelial cells. And what happens is the IgA dimer that we see here with the joining chain gets captured by receptors on epithelial cells called the secretory component. So this secretory component has receptors for IgA molecules, IgA dimers, and they bind together. So this illustrates and shows you that here's the IgA that's bound by the receptors on the secretory component. And the secretory component attaches itself to the dimer and pulls it in to the epithelial cell through a process we know as endocytosis. So the IgA is from outside of the cell, it's going into the cell and being engulfed by the epithelial cell. While it's going to help the entire process, this entire molecule now, the secretory component, is going to help that IgA molecule, that dimer, IgA dimer, move through the cell in a process we call transcytosis. Well, that makes perfect sense, right? If we go to, MedTerm 101, it's moving it through. It's helping to transport it through the epithelial cell. And then it leaves the epithelial cell through the process we call excitosis. And eventually, so it leaves the epithelial cells, and again, this is in the MALT area, and we've attached that really important secretory component. The good news is, as I said on the last slide, that secretory component right here that we've attached through this process within the malt makes that IgA really resistant to bacteria and the action of bacteria. So the secretory form of IgA, now that we have that secretory component there, the secretory component allows the IgA to kind of patrol through the mucosa, and acting as that first line of defense. Remember what we said, it's able to neutralize toxins that are produced by bacteria, and that secretory component keeps the IgA from being affected by this. It also prevents bacteria from adhering to the mucosa. So that's one of the first lines of defense, is just being able to keep that pathogen out, right? Viruses as well, pathogens, it's going to keep, I said bacteria here, but it really is pathogens. We could think of viruses, for example, as not being able to attach to the cell walls of malt there. We know that it's transfers passive immunity to the newborn during breastfeeding. And so it sends some of that, gut health from the mother to the infant. IgA also, the IgA, when it binds, so remember it still has that variable region there, and it's produced by plasma cells. So we have specific IgA that's produced. IgA can bind to antigen, and they get trapped in the mucosa, that entire process, and get eliminated, as we spoke of in the first line of defense, in the innate immune system, eliminated by kind of the motion of cilia on epithelial cells. It also can trigger, unfortunately, a second pathway, 2 out of 3 pathways of the complement system. So this is kind of a redundancy. So if, for example, it's attached to a pathogen that isn't triggering the classical pathway of complement, this is a second way that we can trigger complement, again, to end up with the end result of lysing that pathogen and killing it. Additionally, so you can see IgA is a really important immunoglobulin. IgA can bind to some receptors on things like neutrophils, monocytes, phagocytes, and trigger what we call a respiratory burst, and it releases all the granules within the cells, all of those pathogen-killing granules within the cells. So that's a fantastic thing. And both monomers and dimeric forms of IgA can act as opsonins or promoters of phagocytosis. So that means both secretory, those ones in the malt and serum or serum-based or plasma-based monomers can act as an opsonin. So we're moving away from IgA to another really, really important immunoglobulin class of the immune system. It's the IgM class, and this is the pentamer. This is huge. This is the biggest of all the immunoglobulins because it's got 5 monomers attached by J chains. So it's a pentamer. Represents about 10% of the immunoglobulin pool, but again, theorized that it's not because of its importance, but really because it's got a half-life that's quite, it's a quarter as long as IgG, roughly. IgM is very, very large, molecular weight of 900,000 Daltons. It's big. That thing is not moving through cells through diapedesis, or it's, what I mean, it's not going to squeeze between a couple of cells of the vasculature. So IgM is really required to, it's restricted to intravascular space. The good news is IgM is not super specific. This is the antibody that's the first one that gets produced when you encounter a foreign antigen for the first time. It doesn't have, we haven't developed really great antigen binding sites yet, that site of the immunoglobulin that's going to find that exact perfect lock and key. But there's so many binding sites. So an IgM has 10 binding sites. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 5 times 2, 10 binding sites. So even if it's not a perfect fit, it's sort of a function of, brute force. There's so much there that we can do a great job in the primary or the first immune response that we see when we encounter an antigen. So IgM is a really important immunoglobulin in that first time we see any pathogen. IgM can in fact exist as a monomer on the B cell surfaces. Remember, sometimes we see immunoglobulins on the B cell. But it's generally in pentameric form once it's not in the secretions like we just saw, but once it's secreted by plasma cells. Pentamers are held together by those same kinds of J chains that we just saw, and here's an illustration of one here, how there's one monomer and one monomer being held together by a nice J chain. As I said, 10 antigen binding sites, and it's the first immunoglobulin produced when we encounter an antigen. With its 10 antigen binding sites, IgM is said to have a valency of 10, so it has a very high valency. And so even though it's, as I said, the lock and key isn't a perfect fit, that, kind of force of binding with 10 binding sites makes IgM a really effective immunoglobulin within the immune system. The weak lock and key is a measure of what we're going to talk about within the next 2 or 3 lectures, of what we call affinity, or the strength of the bond between them. IgM has low affinity because it's not a great match. It's not a great lock and key. But once you get 10 of them binding off, the high valency just kind of overrides that kind of weak lock and key bond. So that high valency is super important in the functionality of IgM. As I said, mainly found in the intravascular pool. Why? It can't get out. It's too big. It's also too big to cross the placenta. It's not going anywhere. It can't get out. It's gigantic. This is what we classically know as the primary response antibody, the one that appears first after antigenic stimulation. And also as infants start to develop their own immune system, this is where we see first. The problem with IgM, though, is that it doesn't come from memory. It's really synthesized only as long as the antigen is around. So it's a great first responder, not great for kind of long-term effects, and you can see that the IgM only stays in the plasma for 6 days, so it doesn't have a long life in that. IgM is the most efficient immunoglobulin in triggering what we're going to call the classical pathway of complement, and we'll go through that. A single molecule of IgM can trigger complement very strongly because it has those 10 binding sites. So one molecule of IgM can bind to 10 different antigen sites, and it causes a really strong trigger of that immune process of trying to kill pathogens by lysing them. We are going to see, for a few reasons, it's in the laboratory, in vitro. It's a super good agglutinator. It's also a great method of opsonization. Again, it's really potent against bacterial infections. results in opsonization. And like we've seen before, it can also neutralize some toxins. It's not here, but I would like you to make a note that IgM can, as I said, can be in a monomer, but also we see that it gets attached by those J chains, right? You see those J chains here. The interesting thing about that is that because of those J chains, it can acquire a secretory component. IgM can acquire a secretory component and be a secondary patroller of mucosal surfaces. Let's see. The last thing I guess I would say about IgM here is that the presence of IgM on the surface of mature B cells lets us know that the B cells are in fact mature and they have they've been producing immunoglobulins. We saw that, right? We saw how the mu chains are the first ones that form in the B cell during differentiation, and so when we see them on the cell surface of B cells, we know that we got antibody production. Let's move on to the epsilon chains of the IgE molecule. This is just really minimally in the blood. It's produced by plasma cells primarily of the lungs and the skin. IgE, unlike some of the other ones we've seen before, does not fix complement. IgG and IgM fix complement. IgE does not. IgE also does not cross the placenta. IgE does play an important role in allergic reactions, and we are going to see this when we do an entire chapter on hypersensitivity reactions. What happens is that IgE, when it's produced, can affect mast cells, basophils, and eosinophils, those cells of allergic reaction, and it does it by triggering allergic responses. And how this triggers it, we'll see. I'll explain it now. I don't think you need to know it now, but if we imagine that we have the Y of the IgE molecule, we bind the two antigen binding sites of IgE to a couple of different epitopes on a pathogen, and once it does, that cross-linking that happens results in release of all sorts of mediators, helpers. It releases things like histamine and heparin, which trigger an allergic reaction. We call allergic reactions type 1 immediate hypersensitivity reactions. And again, we're going to spend an entire chapter on hypersensitivity. There are 4 types of hypersensitivity. We're going to certainly talk about type 1. IgE also is involved in parasitic infections by triggering things like neutrophils and eosinophils to destroy those large antigens we saw. In that very first week, we saw how the eosinophils were chasing and not chasing, but drawn to a pathogen, a worm, and we saw it destroying a worm. So IgE plays a role in that. This is just an example of what a type 1 hypersensitivity reaction is. So this is a response to things like allergens, things that cause someone to have an allergic reaction, pollen, hay fever, those kinds of things, peanuts, immediate responses. Some of the things that we see, we'll see things like vomiting, diarrhea, hives. You see these kind of hives, but not to be underplayed. Type 1 hypersensitivity reactions are quite intense, and as a result, they can kind of go overboard. They produce such a strong response, we can have what's called an anaphylactic response or an anaphylactic shock, and that's really a hyperimmune response that can cause some significant damage to the host. Our last immunoglobulin of note is the IgD. These are the delta heavy chains, and as I said, for the longest time, it was the, I don't know, immunoglobulin. It's a single structural unit. It's a monomer. Very, very small percentage in the immunoglobulin pool. We see it now on unstimulated lymphocytes, and so we really think now that primarily research has shown toward it is demonstrating an ability to extend the life of a B cell. And it's also known to aid in B cell activation. But this extension of B cells, when you can bind IgD, when IgD is bound to the B cell, that's an important factor. Remember, we find these on unstimulated lymphs, so we don't want to have to be producing things all the time. IgD, just like pretty much everything except IgG, does not cross the placenta. Only IgG crosses the placenta. Only IgM and IgG will fix complement. Very short half-life on IgD itself, though. So let's look quickly at humoral immune response production. There's a couple of phases of antibody production. The first phase is the primary phase, and think of that as the first time we get exposed to an antigen and how the immune response develops. We're going to see in just a second. Actually, let me go ahead here, and you can kind of go back and forth between the two slides when you're doing it. The first exposure to an antigen, during that period of time, the immune response has what we call a lag phase. It means that there's really, so if this is the point of exposure, and this is the time since exposure, and this is how much antibody we produce. During the first, 5, 7, 8 days, we're really not producing virtually anything. We're producing no IgG for sure, but we're kind of gearing up the immune system. At around the 8-day mark, 7 to 8 day mark, we start to see levels of IgM in blue. IgM is the first one to respond, and you can see it's just shortly ahead of IgG in production. It's a few days ahead. So IgG, that not so specific but really potent pentamer, is produced first. And during that first phase, we get kind of a gradual rise. So if we look, this gradual rise, honestly, by the time it peaks, by the time IgM peaks, we're at about 15 days, and by the time IgG peaks, we're at about 18 days. So it takes a good solid couple of weeks before we get kind of our peak rise of antibody production. This is why that the innate immune system is so important. Being able to produce immunoglobulins is not our first response. It just isn't. In the initial time we see an antigen. After, in that roughly, 2 to 3 week, just under 3 weeks, 18 days, let's say, 2.5 weeks, it plateaus and those immunoglobulin levels drop off. So we see them stick around for a couple, 15, 18 days. Antibody production lasts about 10 to 12 days once it starts producing. And then immunoglobulin IgG produces only for about 4 days. It doesn't have a huge amount of production time. As long as there's no further antigenic challenges, we're not, the antigen has been killed off and we don't have a recurrence of the infection, those levels drop off. IgG may continue to be produced even after we stop seeing the antigen, but in very small amounts. So you can see IgG kind of tails off. The second phase of immunoglobulin response. So let's say we had the first exposure, we had that peak, we had a drop off, And then at some point in the future, we had a subsequent, so we had stuff sticking around. Remember, IgG will stick around for 23 days, right? So we have a period of time during which we have immunoglobulins in our blood and tissues. And if during that time we get a second immune response, we get a second contact with the antigen, that's when our memory kicks in. And remember, we talked so much about the production of memory B cells and memory T cells. In this case, those memory B cells are either going to be activated directly by the antigen, in that T-independent one that I talked about in the very first slide, or the T cells are going to are going to be going through, there'll be antigen presentation by cells with, depending on what it is, whatever the antigen presenting cell it is, and giving it to the T cell, and those T helper cells are then going to trigger the production of the activation of the B cells, right? So that's one of the responses of T helper cells, is it can trigger the B cell production. So, but we've already seen it. So the subsequent time after we get contacting the antigen the second time, look at the IgG response. So here's the response here, and instead of it being 8 or 10 days later, it's days later. But at the exact same time that IgM is kicking in, to again just about the same level, IgG, that really strong responder, kicks into overdrive, and you end up with super high levels of IgG immunoglobulin production that responds very, very quickly and very strongly to that second response to the antigen. This is the process we call memory. This is the memory production. This is the initial production. This is the memory production. We call this the primary response, or first response to the antigen. This is the secondary response to the antigen. So, as I said, if we compare the second time the host encounters the same antigen, IgM and IgG lag time gets shortened considerably, though synthesis is just about the same from IgM. IgG, though, has a really marked and much stronger response to a secondary exposure. So in both cases, the response is quicker. The IgG, the response is much, much stronger, though. And in fact, in a secondary immune response, we consider IgG to be the primary responder of immunoglobulin in response to a secondary exposure. This is based on the fact that we have memory cells. We've already seen that antigen before, so we recognize it, and the response is immediate. The immediate production of those protective antibodies are primarily IgG. You still see some IgM, but when we're looking in the laboratory to determine if someone has been exposed to something before or not, we're going to look specifically to see how much IgG they have. If they have a very low volume that's about equal to the amount of IgM, we know it's the first time they saw it. If they have a very strong response that's considerably higher than the amount of IgM, we know it's a secondary response. I should mention here the secondary response is also called the memory response or the anamnestic. So it's ANAMNESTIC. I'm actually just going to type this in for you here so that you can see. So it's the, oops, it's not that easy to type here. Memory, or anamnestic response. So this is also known as, okay? You should know those two terms as well. So I'm gonna highlight the whole thing in red. You can figure out that I don't really mean you need to learn the word or. Okay, let me do this and move this along. I'll take this off and, all right, there we go. All right, awesome. Back, there we go. So it's the memory or the anamnestic response. So we've seen the production of antibodies. We've seen what happens with the first and second responses. How do we control it? It's really important that we control the immune response as well. We can't have just unfettered responses to everything in kind of an overwhelming amount. We'll see that those kinds of responses can be quite harmful to the host. So we have a complex series of controls of the immune response, and the concept of these immune responses were based on a couple of premises. First is that, just genetically, some species are really good antibody producers and some aren't. Antibodies produce very high levels of, rabbits produce very high levels of antibodies to proteins, mice don't. It's just a function of their status as, a host or an individual. But what's really important is the control of the immune response based on genetic diversity. And that is that humans don't make massive amounts of immunoglobulins unless it's really in response to a specific antigen, right? So we're not off producing high levels of the antibody every time we see, we don't trigger every single potential antibody every time we see an antigen, right? So they're based on the fact that we can produce anywhere between 10 to the 6th and 10th to the 9th different antibody molecules in our lifetime, all individualized and all based on the genetic control of antibody diversity and being able to recognize different kinds of antigens, all through that processing, through the MHC complex, through antigen presentation, as well as the variability of the those really minor changes that we spoke about within the immunoglobulin barrel, that hypervariability region. So we can make a lot of different antibodies in response to recognizing a bunch of different antigens. There are a couple of hypotheses about how antibody diversity came to be and how it is. I think most people agree now that kind of the clonal selection that we're going to talk about in just a second is the one that is thought to be most accurate, but there is one just for the sake of kind of historical understanding called the Ehrlich's side chain theory. And in this case, the theory was that really some of your immune cells had receptors to the antigen long before the contact ever occurred. They were kind of pre-made cell receptors. I think now we have figured, now we understand how antigen receptors are made and how presentation affects that. So I think that we can ignore that for the most part. Clonal selection, on the other hand, proposes that lymphocytes are genetically predisposed to produce one type of antibody, and that antigens actually, in a way, select those cells that are capable of responding to it, and that it's by sort of the action of the type of antigen that it is dictates what cells respond, and that in fact that because we have that kind of pre-known idea of what kinds of cells are going to respond best to different kinds of bacteria or viruses or other pathogens, those are the lymphocytes that proliferate in the greatest concentration when we're exposed to it during those exposures. And then I think that now we recognize in a way that genetic coding, the more we understand and unravel the genetic coding of immunoglobulins, it's thought that genetic coding in cells looks at those three clusters, looks at three clusters within cells, and the gene rearrangement that happens during cell differentiation ends up resulting in immunoglobulin synthesis in direct response to those genetic codes for the production of immunoglobulin. So gene coding is, I'm not really concerned about that. What I'd like you to know for definitions really are the Ehrlich side chain and the clonal selection. Those are the two most researched, I think, hypotheses about how we get antibody diversity. And so that's it. We're done. I am going to be sending you a little bit on the next chapter that we're due to cover, but this will take care of about half of your lecture for the week. So be on the lookout for one more lecture of just a little bit under an hour for Tuesday's online lecture. Thanks so much. Ask me any questions you have. Send me an email. Send me a message, or just go from there. I also just want to point out to you, there's a description of, so here's some study guide stuff, but here's the instruction for making the immunoglobulin table. Here's how you do it. This is what needs to be done. This is an example. This may not work for you. I don't care if you don't do it on a spreadsheet. Some people draw pictures of them. Some people, I've seen these done in all different mechanisms, and other people have added other categories in here that were important to them. So remember, this is a study tool for you to have really a big picture of all of the immunoglobulins in one table, something that works for you. Remember, make a copy of that for me. Don't give me your original. That's going to be a study tool for you. Okay? Sounds good? Awesome. I'll see you next time.